In this second micro lecture, I wanted to take basically new evidence for the developmental origin of health and disease. What we reviewed in the first micro lecture was basically early studies, clear epidemiological evidence. I want to go now in a little bit more detail and basically show to you that the idea of developmental origins of health and disease applies to many, many different conditions. Notice that the developmental origin of health and disease have been so influential that it even appeared in the main cover of the magazine Time, for example. But this has been quite common. There's lots of popular programs more now, and you can even see this on TV. Developmental origin of health and disease on or effects on reproduction. One example here: exposure to diethylstilbestrol. Well, exposure to this DEB, let's call it simpler, DEB in utero, increases the incidence of vaginal adenocarcinomas at puberty and breast cancer. Why would that be relevant? Well, what we know is that DEB is actually a synthetic non-steroidal estrogen. It's an endocrine disruptor. The point is that between 1940 and 1970, DEB was actually given to pregnant women to reduce pregnancy complications and pregnancy losses. So this was a common drug, a common medicine that was given until it was realized that actually the offspring, the children born from mothers that actually had received DEB were called DEB daughters and DEB sons because of the problems incurred. In mice, it's known that DEB increases the incidence of adenocarcinomas, not only in the first generation, not only in the offspring of mothers that have been treated with DEB, but also in the second generation. What would that mean? That basically would suggest the existence of transgenerational effects. This is quite common, and this is one of the main studied areas in the developmental origins of health and disease. Exposure to drugs or toxicants or substances early in pregnancy will have large effects later on in life. The proximal mechanisms that we interpret are involved here is basically the alteration of the estrogen response at puberty. It seems that children whose mothers basically were treated with DEB will show a differential or an altered estrogen response at the time of puberty. And this is seen late in life, not during childhood, but late in life or later in life, during puberty and later, basically there is a larger onset of adenocarcinomas and even breast cancer. Of course, this details that these effects can be transgenerational calls up for another question, and that's the study of the long-term mechanisms. How could this be inherited? How is it possible that if your mother, or put it this way, if your grandmother was treated with DEB, you, the grandson or the granddaughter, actually are exposed to these effects? So we are talking about effects that are very long-term, for which we know very little the mechanisms. Not only this, effect of the developmental origins of health and disease on neurological disorders. This is very popular nowadays. I found this table in a paper in which basically they relate the different neurological conditions, such as increased anxiety, increased ADHD, conduct disorders, low cognitive performance. This is actually related with prenatal stress, with stress at the time, stress from the mother at the time of pregnancy. I must say that most of these studies are quite controversial, because we have very little knowledge at the moment of the mechanisms that are involved here. It's very easy to say that a lot of disorders that one observes and whose incidence increases in the general population are actually due to prenatal stress. We cannot discount that either, but you can be for sure, you can be certain that this is going to be an area of heavy study. How is it is it possible to characterize the mechanisms that explain that stress during pregnancy leads to an increased risk for ADHD or other anxiety or other neurological disorders. But I think that it's important here to reflect that the proximal mechanism in this case expected to be is that the alteration of the HPA axis reactivity. Remember we talked earlier in the course about the HPA axis, the hypothalamo-pituitary adrenal axis responsible of stress. Of course, prenatal stress could affect this HPA axis and program this for later in life. We know, for example, of the existence of epigenetic alterations in the glucocorticoid receptor gene associated with this. But, I mean, we know relatively little about these processes. It's a work that is actually ongoing. We know, and this is also an area being increasingly recognized, of the impact of assisted reproductive technologies on later health. Assisted reproductive technologies means situations in which fecundation does not take place naturally, and there is an assistance in the form of in vitro fertilization procedures or tube babies. Any kind of situation in which you deviate from the regular fecundation, natural childbirth, actually nowadays it's being recognized that it can incur in some problems. And this is not a small number. 1 to 3% of all births in many developed nations are actually the result of one form or another of assisted reproductive technologies. It's known that ARTs are associated with multiple births. That means that it's not only one child, but basically 2 or 3 or even more. It's also associated with low birth weight, a reduction in birth weight, and there is a larger probability for major malformations. So ARTs, assisted reproductive technologies, have been very successful, have given a lot of couples the possibility of have biological children, but we are still starting to understand the long-term consequences for these type of procedures. Notice, and this is relevant, that ART, assisted reproductive technology, involve a lot of processes that are not equally natural. For example, you start with ovarian hyperstimulation. Mothers are stimulated, so they ovulate more, so that the oocytes can be collected. They can be incubated with sperm, or of course sperm can be injected. Then they are cultured. These oocytes are cultured externally from the body for up to 7 days. For up to 7 days, depends on the age and depending on the procedure, but you have then the zygotes that will be from the blastocyst stage to 6 or 8 cells, or up to 100 cells if it's a later type of zygote. And then it's this oocyte is implanted, or not. In some cases, these oocytes are frozen and later on thawed and implanted. So of course notice that there is a lot of steps that you are subjecting to the oocytes and also the sperm that are artificially outside of a purely biological scenario. So of course there is the danger for most of these or many of these procedures. We don't know exactly the consequences, the effects that they can have on these cells. Of course, it makes it even more dangerous the fact that assisted reproductive technology occurs at a critical period of development, the very, 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 very start of development. Not surprisingly, you should know that only 15 to 30% of the treatments actually result in a live delivery. So there is a high degree of failure, which already tells you that there's lots of things that are not totally controlled. In an interesting study from a couple of years ago, this group of Swiss researchers basically compared babies born from assisted reproductive technologies with control babies. And they did that well, they were not babies, they were children at 12 years of age. And they compared basically the changes in the blood flow to the arm in these children and compared them. What they observed, for example, is that flow-mediated dilation in the brachial artery, flow-mediated dilation is decreased in ART children. You are going to say, well, what does this actually mean? We don't know. At this point, we don't know. But the suggestion seems to be that in ART babies, vascular reactivity is affected. They also see, for example, that the pulse wave velocity in the carotid artery is slightly enlarged in ART babies or ART children, and the intima to media thickness of the carotid artery, that means how thick the vascular wall of the carotid, is also thicker. So there seem to be some vascular differences, vascular reactivity differences between control children and children from ART procedures. We don't know yet if this can have long-term consequences, but there is a difference. And finally, the effects. I want to talk about the effects of hypoxia on the developmental origins of health and disease, in particular looking at the cardiovascular system. We know very well that hypoxia causes IUGR. IUGR stands for intrauterine growth restriction, and it also causes low birth weight. Of course, no one is going to be surprised to know that the economic status also affects birth weight. And that's because basically there is a direct link between economic status and maternal undernutrition. Poor mothers usually cannot afford to feed equally well, and there is a malnutrition during pregnancy that can have long-term consequences. When you combine this with hypoxia, there is some interesting results that have been published. These results have been obtained in Bolivia. And what you see here from the study by Giussani is the two sets of curves, or actually four sets of curves here. The black closed symbols are actually results from the city of La Paz. The city of La Paz sits at 3,500 meters, so it's high, relatively high altitude in a little bit hypoxic conditions. And then you have the two curves here. What do these curves show? It's basically birth weight by percentage. So the percentage of babies that are born at a given weight. When the curve is shifted towards the left, your left, it means that there is a higher percentage of babies that are born smaller, right? So what's interesting here is notice that the baby born in La Paz, these two lines, are smaller in percentage terms. There are more babies that are smaller than babies born in the city of Santa Cruz. Santa Cruz is a city, Santa Cruz de la Sierra, that is sitting close to the jungle, basically, at much lower altitude in Bolivia as well. So basically, babies born in La Paz are smaller, in this curve is shifted to the left, than babies born in Santa Cruz. But what these figures are also showing is something else. It's showing social or economic status. The circles are babies born from high-income families. The squares are babies born from low-income families. And what you see is that in the city of La Paz, the families from the high-income families actually give birth to smaller babies than the low-income families. But this trend is reversed in Santa Cruz de la Sierra. So what you see here is that at low altitude, basically economic status kicks in, and babies from families that have a low economic status are smaller. But in high altitude, or at the city of La Paz, it's the opposite. So at the city, in the city of La Paz, it's not economic status that matters, it's adaptation to altitude. Because there is a clear connection between poor mothers and Aymara Indian ascent. So most of the poor natives here are actually descents of Aymara natives that have been living at altitude for a high. So this is what has been called actually the Andean curse on the conquistadors. So basically the people that moved to these areas as conquerors, the Spaniards that moved there, suffer from these much more than the natives that are there as a result of altitude. So in this case, the outcome is that you are bound to expect a higher incidence of diseases in babies born at altitude as a result of the hypoxia during development. Why is that so? Well, there are many different mechanisms, but according to Giussani, who has done this study, his main bet is for catecholamines. Basically, the idea that hypoxia triggers an increase in catecholamines, an increased peripheral vasoconstriction, and what is called asymmetric IUGR, intrauterine growth restriction. It's known, we know very well, that hypertension during pregnancy is also higher in mothers from European ascent. And that's what you see here, basically. Mothers from European ascent or Andean ascent, basically this is your genetic background. What you see here is at altitude, the black bars, the European mothers have a much higher systolic blood pressure than the Andean mothers at altitude. The Andean mothers, they distinguish very little at low altitude or at high altitude. But in the European mothers, there is a very large difference. So we don't know yet what are the causes, but the expectation here is that there is a difference in oxygen delivery through the placenta during the time of pregnancy associated with vascular abnormalities. To summarize this, I think that what this evidence is giving us is that the prenatal and the postnatal environments are important. We already knew that the postnatal environment is important. We are bombarded with information telling us that we need to exercise, that we don't need to smoke, that we don't need to drink too much, blah, 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 blah. We know how important that is. That's the postnatal environment. What this data is showing is that the prenatal environment is also equally important. And you can say, well, there we have no choice because we can choose very little or nothing. It's our parents that have to choose for us at that time. The developmental origins of health and disease is not only associated with low birth weight. This was the start. That's the start for Barker's hypothesis. Nowadays, we recognize many other risk factors that are not uniquely associated with low birth weight. Put it in other ways. Are you born normal for size? Does that mean that you cannot have any problem? Obviously not. There can be other risks. But what the original studies pointed out was that low birth weight was important, but there are other factors that are important. Finally, the developmental origins of health and disease reflect on a third factor in the etiology of many diseases, on the origin of many diseases. It's not only important that you have a good gene pool that basically makes you healthy, it's not important that you behave well as an adult, it's also equally important that during pregnancy things went as they should have gone. So that is what I call the third factor in the etiology of many diseases. And notice that this third factor was recognized by David Barker in the 1990s, basically. So we have a much shorter 30 years period of study for this type of complications in comparison with the understanding of genetic diseases, the understanding of environmental factors that affect disease. And that's it. Thank you very much. Time for questions.